Hello. We'll wait for some time if someone joins in, or else we'll start the lecture and the recording will be available. Okay, we'll start with the lecture. Share the entire screen. Okay, so this is the course for Bioengineering and Interface with Biology and Medicine, which is an NPTEL course by Professor Sanjeev Srivastava, and I am the TA, second TA for it. We are two TAs for this course, and one of the TM had already took this week's overview lecture on Saturday, and now I'm here to for few of you to just revise it on Tuesday. So this is the week 5 lecture of this course, which is by NPTEL itself. And so, sir, what majorly talked about? So there were 5 lectures in week 5, and sir majorly talked about how. So in the initial lecture, we talked about how genetics, how inheritance happens from one generation to another generation as per the Mendel's law as well as non-Mendelian inheritance pattern. But there were few other scientists who tried to check whether there are chromosomes involved in inheritance pattern or not, like how exactly chromosomes are a part of it. Uh, so the that was something Sir talked about. Then we talked about how like the same experiments, the one which were done in plants by Mendel, some similar kind of experiments were done with Drosophila. So when that was accessed, like that was tested and tried to determine how inheritance happen uh, in an animal basically, and how in happens in plant, and as well as all those characters which generally Mendel dealt with were autosomal characteristics, whereas um, there were some characters which were learned by other scientists which were related to the sex chromosomes, how like a male can transfer it to daughters only or a female can transfer it to sons only. So like we'll be dealing with all of those stuffs in this week's science lecture. So all those were the major part of the whole five lectures in terms of week five. So let's have a quick revision on that, and then we can move on to understand oh, sorry. Then we can move on to check the previous year questions assignments. Okay, so to give a recap of the initial lecture, so as I told that it deals with the chromosomes are involved in it. So like to give an assurance that yes, it is coming from the chromosomes. So as we already talked about dihybrid cross in the last lecture, wherein a yellow round seed was crossed with green wrinkled seed, and how what exactly happens and how exactly it is getting like transferred into the next generation. There might be, so it happens because of meiosis, which is indicating that they are assorted separately, uh, all those characters, whether it be yellow one which is the dominant gene, and green one which is the recessive gene. So recessives are coded here with in blue and dominant are coded here in red, and how these chromosomes basically get assorted, and two different characters. This R is for round, and this small r is for wrinkled, whereas those two different characters of basically two different traits of a specific seed, how they are independently assorted, all those were proven. Like all this kind of system we actually see in meiosis part of cells. So this is the chromosomal basis of Mendel's law. So if you see the law of segregation where two alleles of each gene separated, so this of the two alleles of each gene, so this is the each gene of yellow, and they are separated together. If you can see here, RR are separated, Y is separated, as well as these two are separated, and similarly they are independently assorted as well. They are not dependent on each other. Both characters are independently assorted. That's what we can see in our meiosis thing as well. So that's what Sir informed, like Sir was talking about. And next slide, please. So Sir majorly talked about the chromosomal basis of Mendel's law, wherein so that was the conclusions which we got from the initial part of the lecture, wherein he talked about results of Mendel's dihybrid cross, and he that was correlated with the chromosomal behavior as compared which was shown in terms of meiosis. So one of the scientists actually tried to prove it that it happens because of meiosis, whatever Mendel saw in terms of dihybrid cross or monohybrid cross. So this behavior of chromosomes during meiosis in F1 generation and subsequent random fertilization give rise to F2 phenotype ratio, which was observed by Mendel. So the observation which was happened, so Mendel majorly dealt with what is being observed, whereas uh, this specific scientist who is Morgan, he did certain experiments to tell about that genes are associated with specific chromosomes. So that was his experiment. So this is how wild type eye generally looks, and the mutant eye of a Drosophila is white in color. And how do we donate like denote them in terms of crosses or in terms of their uh, alleles? So for a dominant gene, a red eye, we generally denote with the plus sign, which is the wild type, and the mutant type, which is white. So that is without the plus sign. So that's how we denote it. And so if we do a cross between a wild type red-eyed, so that is W plus female, mating with the mutant white-eyed male. So this is white-eyed male and this is the wild type red-eyed female. So if we cross them together, if we, and the results which were got for all offsprings had a red eye. So this, if you remember the Mendel's experiment, so this is similar to that of a purple flower is being crossed with a white flower, and as purple was dominant, in the F1 generation, all the offsprings were purple in color, although they were carriers of it. A similar kind of experiment was performed in Drosophila, and it was similar results were obtained in terms of Drosophila as well. And so if you draw a Punnett square board of it, so this is how it will be. And so what will be the F2 generation? So in F2 generation also, if you remember, in terms of monohybrid cross, like take dealing with only one specific character, that is red eye or a white eye. So when they were crossed, these were carriers, so W plus and W, and male was also a carrier W plus only. So when they were crossed together, we could find three red-eyed Drosophila, whereas one which was 3:1. So this is a similar ratio which we also get in terms of Mendel's experiment. Sorry. So if we talk about dihybrid process between them, that is a mutant white-eyed male, uh, what color eyes will be the F1 and F2 offsprings have? And we actually found that in the earlier slide that when F2 generation had with the carriers of male and female, we generally get three red-eyed Drosophila and one white-eyed one, which is similar to that of Mendel's experiment. So that was interesting to see that a recessive trait white eyes was expressed only in males in F2 generation, else it was not in females because the other female who had a uh, uh, like W plus gene, so that's why they were carriers of it, but they were not affected with the color. And therefore, Morgan concluded that eye color gene is located on the X chromosome and there is no corresponding locus on Y chromosome because it was affecting only males. And if it had in the females as well, but the other gene of the female is the with the W plus characteristics, that's the reason they were carriers, but they were not affected. So that's the reason no females were affected with this specific color change in terms of eye, only males were affected. So that is what Morgan concluded, that experiments provided evidence that chromosomes are indeed the location of Mendel's heritable factors. That's what Mendel tried to prove. And so, in terms of chromosomal basis of sex in humans, so we have 44 chromosomes along with sex chromosomes, wherein 22 were autosomes and 22 were so along with 22 autosomes, one is sex chromosome X and one is the sex chromosome Y. So in terms of females, there are two X which are sex chromosomes, and in terms of females, sorry, in terms of males, it is X and Y, which are the two sex chromosomes. So only males are having Y chromosome. Females don't have Y chromosome. So that's the reason sex of an offspring depends on whether a sperm cell contains an X or a Y chromosome. If a sperm cell contains Y chromosome, then a person or a individual or a next generation which is being born will be male, and if it is an X chromosome, it will be female. So what do you mean by sex-linked inheritance? So sex-linked gene is nothing but a gene which is located either of the sex chromosome, either X or a Y, and Y-linked genes are the one which are located specifically on Y chromosomes. And there are about 78 genes in Y chromosome which mainly help to determine sex. If it is Y, then it is a male, and if it is X, then it is female. Whereas in terms of X, there are 1100 genes involved, and that gives responsible for many characters, but unrelated to sex. So Y is the one which actually determines the sex. So if there is no Y, then could be female, and if there is Y, then it could be male. And if you all can read this, then it indicates that you all are not colorblind. So this is one of the trait which Morgan tried to study. Not exactly Morgan. Morgan tried to study all those characters in Drosophila, But the various scientists tried to study where sex-linked traits, and one of the trait was color blindness. Like if it is there in the female, then how it is getting transformed. If it is there in the female, then how it is getting transformed to the next generation, whether it is going to only females or like daughters or only sons. Like how exactly that pattern is there. So they tried to check those patterns, and different kinds of characters were studied: red green color blindness or night blindness or hemophilia or muscular dystrophy. Different kinds of disorders were tested, and which are sex linked basically. That's what it was found. So how exactly they are represented? So a female has two X chromosome, male has one X and one Y. So in terms of females can be carrier because these are colorblind. Red-green color blindness is a X-linked disorder, whereas males are having only one X chromosome, and if that is in a mutant state, the male will be colorblind. Whereas in terms of females, they can be carriers also. So these were three different, which sir explained on board as well. So, but if you just looked at it, so if there is a cross between a normal female and a colorblind male, then if you do a cross Punnett board square, you can easily understand that there will be two females who will be carriers of it, as they are having only one. So the disease one is indicated by small n, and the normal one is indicated by capital N. And if you see in terms of this, if this is how the process, a colorblind man cannot transfer it to a man because its X chromosome is coming from the female, and whereas the female was completely normal, not a carrier. But when if you cross with a carrier female, it can also transform its specific disease of color blindness to males as well, and one of the female can be carrier. So there are different chances. The percentage will be different. Here, 50% of chance that each daughter will be a carrier, and there are 50% chance that each son will be a with a disorder. Whereas in terms of this, a colorblind father cannot transmit it at least to uh, like they cannot be making any male as a colorblind only. It can make all daughters as a carrier. And similarly, when a cross was done between a carrier female and a colorblind male, then there are more chances that 50% of the chance that each child will be having a disorder regardless of the sex. Maybe it be female or may it be male because she itself was a carrier. And if these two X goes into the daughter, then the daughter can also be colorblind. So that's how sex genetic counseling happens. So these are some. This acts as one of the example wherein we can understand how uh, a specific trait gets transformed or gets inherited from our parents to the next generation to us. So that's the reason we have to study all those characters, and we can predict that the next generation, how much percentage of chance, like we cannot predict who will be coming, but we can what characters will be the individual will be having, but we can predict the chances of a specific character which can go in the next generation. So that's how it helps to understand. So fathers, so in order to conclude what how that happened in terms of color blindness, so father can pass X-linked alleles to all their daughters, but to none of their sons, because X is only going to, whereas father gives Y chromosome to the sons, that's the reason only it can be passed to all their daughters, whereas mother can pass X-linked alleles to both sons as well as daughters, because that X will go either to the son or either as well as to the daughter. Whereas any male receiving the recessive allele from his mother will express the trait because the X is only one in terms of males. That's why it will be expressing. Whereas, therefore, far more than males than females have X-linked recessive disorders. So that's the reason that is the case in terms of X-linked disorders. So there was one more disease which is called as hemophilia. So absence of one or more of the proteins required for blood clotting, that is nothing but hemophilia. And you might have heard about this royal family of Europe, wherein who are from Queen's Victoria of England. So that family actually passes hemophilia trait to uh, all its generations since the start. So there's a great story regarding them. and how that specific factor is running in their family. Similarly, a genetic basis of a mutation and how it's resulted in a non-functional blood clotting factor is now understood because of such kind of studies and such kind of process. And people try to understand how hemophilia is getting transformed, like inherited from one generation to another. So just to remember certain points like how this sex-related inheritance happens. So sex of an individual is often chromosomal based, either X or Y. They will actually determine it. And humans are other mammals have an XY system in which sex is determined by whether a Y chromosome is present or not. If Y is present, it will be male. If not, then it will be female. The sex chromosomes carry sex-linked genes. Virtually all of these are wait a second, I just there's Google Meet. Oh, it's logged out. I need to join this again. That's what I thought. Sorry, I was some network problem. Yeah, good to go. I hope I'm sharing the entire screen. Yes. So we were here understanding certain points to remember in terms of sex-linked inheritance. So any male who inherits a recessive X-linked gene from his mother will express the trait as we already discussed about it. And in terms of mammalian females, one of the two X chromosome in each cell is randomly inactivated during early embryonic development. So in order to conclude the first lecture of uh, the fifth week, so he talked about Mendelian inheritance and chromosomal involvement in terms of how inheritance happened in terms of segregation and independent assortment, and Morgan who tried to give a chromosomal basis of inheritance as per Mendel heritable factors, and also talked about sex-linked inheritance, whether it is X, Y, how exactly color blindness, hemophilia gets inherited. Then we came to the second lecture where the dihybrid uh, process of Drosophila were explained, wherein two specific characters were considered, whether how what is the body color and how uh, the wings are of Drosophila. So in terms of wild type, if they are having a gray body, so gray body is generally the wild type one, whereas black body is recessive one. And generally, Drosophila have good normal wings, whereas some of the Drosophila have vestigial wings. So that's how we are donating. As we already talked about it, so dominants are denoted by a plus sign, and um, the mutants are given no sign as such. So this is for body color, and this is for wings, which is vestigial wings or normal wings. If it is a plus sign, it's a normal wing. So this is how the cross will take place. And if you are crossing uh, uh, wild type with double mutant, like which doesn't have either normal wing, either a gray body, which is of a wild type characters. If you cross them, it is completely recessive, double mutant, and it's completely dominant. Uh, when you cross them, then a feature which comes in. So phenotypically, it is remaining as gray body and normal wing, but they are carriers of it. If you can see the genotype of it, it is one plus and one with no sign. So this indicates it is a carrier for body color as well as for wings, but phenotypically it's looking the same. So that's how we were also dealing with in terms of yellow colored seed, which were actually carriers when we crossed with the dominant yellow round with green wrinkled. So kind of if you can link with that, so it will give a similar result as which we got for uh, uh, yellow round and green wrinkled seeds. So when we cross them, we will get a carrier one, and when you so this is talking about the test cross in order to determine the genotype of this specific of F1 generation. So that for that you need to cross it with the recessive one, which is which is with the black body and vestigial wings. And when you cross it, the characters or individuals which come in. So somebody joined it. Okay, Sham and Unnati joined the lecture. Thank you for joining. I actually started the lecture at 7 itself, but in between, I think I got logged off because of some network issues. But we can continue. So basically, I'll initially giving a recap of the lecture 5, like week 5 lectures, and after that recap, we can go through previous year assignment questions. Yeah. So when you, in terms of touch crossed, as we already learned about it, that we need to cross it with the recessive individual, the one which is showing those characters. So when we cross them, it will try and let us know what is the genotype of the uh, individual which we are trying to determine. So if this is how the characters are appeared, like with a gray bordered normal wing and a black bordered vestigial wing, black bordered normal wing, as well as gray bordered vestigial wing. So all different characters. So if you see here, these were two characters phenotypically appeared, but here two different phenotypic characters were also came into picture. If you see, so these two characters were already there. These two characters came into picture, which indicated that this is a carrier and not the same. If it was same like this, then all the offsprings would have been phenotypically normal. All that they would be have been a carrier, but phenotypically looking normal with gray bordered and normal wings. But if you when it was crossed with the recessive one, you can see two different characters, which indicated that this is a carrier and not a, uh completely wild type with black bordered and normal wings. So that was actually talked about it. So how does linkage between two genes affect inheritance of characters? So most offsprings had a parental phenotype, if you see here in terms of this, but uh, relatively smaller number of offsprings with non-parental characters. So this is talking about these are parental characters and these are non-parental characters, as you cannot see such characters in their parents. So, which indicated that there is some mechanism occasionally breaks the linkage between specific alleles of genes on the same chromosome. So it is on the same chromosome, but if you see at the characters, it is changing. So that is because of recombination, which will come into picture in the later slides. So alteration of chromosome numbers or structural causes some genetic disorders. This is some related to how the number of chromosomes, if there is a change in that, how it exactly affects an individual. So we generally study that using karyotype. So karyotype is a technique wherein uh, we take a cell and we try to visualize a chromosome, and this is how initially a chromosome is visible. Then with the help of karyotyping, we actually select each one of the chromosome and align it as per the uh, so these are homologous chromosomes, right? So in this specific, all 20 to 44 chromosomes will be visible, wherein 22 are autosomes and two are two are sex chromosomes. So according to the sizes of the chromosome, we can actually arrange them like this and give a specific color as well, which will indicate chromosome 1, 2, 3, etc. So that's how karyotyping is done in order to understand. So if there is any chromosomal aberration change in the chromosome number. That's how generally people determine whether it is if there is a change in chromosome number. And so just to understand few terms related to alteration in terms of chromosome. So if you talk about deletion, so if these are the genes involved in it, so if there is a deletion, this one deletion removes a chromosomal segment. So these are different segments. If you deletion indicates that one of the segment is deleted. What do you understand by translocation is, so some part of the chromosome has been shifted to some other chromosome, and this part of the some one another chromosome has been translocated to some other chromosome. So this is the shift. So this indicates that a translocation moves a segment from one chromosome to a non-homologous chromosome. So these are homologous chromosomes, MNOPQR, and this will be. So this is basically to make you understand in a easier manner, but this is not how it is coded. So basically, some part of one of the chromosome, one homologous chromosome goes and attaches with some different chromosome itself. So it can be any number, like maybe a number five chromosome, a part of it might go and attach with the 13th chromosome or something, and so 13th chromosome part of that chromosome goes and attaches with the fifth chromosome. It can happen any which ways. That's called as translocation. And because of such kind of different change in chromosomal structures, different kinds of diseases come into picture. So this is one of the abnormalities which are called as aneuploidy, wherein we are talking about the chromosome number. So this all these diseases are dealing with autosome chromosomes, not X and Y. So there is one X and Y chromosome or else sex chromosome, either 2X or one X and one Y, or else, and whereas these two autosomes are the ones. So if you look at these specific diseases, Turner syndrome, sorry, Down syndrome, Edward syndrome, and Patau syndrome. So these three are related with autosomes, but the Turner syndrome is related with the sex chromosome. So this is the chromosome abnormality that happens when there is absence of one or both the copies of X chromosome. This happens when trisomy of 21 indicates there will be three chromosomes of number 21. So generally there are two, right? We have homologous pairs. So if there is one more accidentally, it's called as trisomy 21. The disease is called as Down syndrome. And similarly, if it is on the 18th chromosome, it's called as Edwards syndrome. And if it is similarly on the 13th chromosome, it's called as Patau syndrome. So you might have looked at like seen this specific movie wherein this specific individual was having progeria syndrome. This happens because there is a mutation in one of the gene called as lamin A gene, and there are currently tests available to identify that, and we can identify that whether the offspring which will be coming into the world, whether it has any kind of mutations or not. So if you might have heard about uh, the genotypic study of the individuals who are actually in the womb of a mother, and people try and test such kind of all of these aberrations and determine whether, so currently we can determine all of those stuffs whether uh, he is genetically normal or not. And in terms of chromosomal aberrations, it's not exactly if there is a gene mutation or not, that all we cannot determine at that specific stage. But if there is aberration in terms polyploidy nature or aneuploidy, if that is happening, then we can determine at that stage as well. And if then we can accordingly abort it and instead of allowing them to come to the world, because that it is difficult to survive with these such kind of syndromes. So how is that happening? So polyploid is a complete set of chromosome in multiple numbers, so either 2n, 4n, or 6n. We are the one who have in 2n. So if there are six chromosomes, if there are three pairs of chromosomes, so overall six chromosomes. So this is how the division happens. Then it becomes a tetraploid cell. So it gets multiplied and then it becomes separates into two different cells. So that is how uh, a cell is dividing basically, and there is a possibility that they two can join together and it can make 4n instead of 2n. Generally, the wild type is 2n, and because of certain changes in the cell division pattern, it leads to 4n, and that's how trisomy and everything comes into picture. So similarly, talked about different watermelon as well as rice grains. So just to check the concept, so determine a sequence of genes along with the chromosome based on the following recombination sequences. So if AB was there, which was around 8 map units, so imagine a chromosome. So if I can draw here something, pen. So imagine a chromosome like this. So if there is A and B, like let's first make it only if there is A and B, which is 8 units, like distance between those two genes specifically is 8 units. And if you see A and C, so we need to make C somewhere far enough, which has 28. So this specific whole region has 28 units. That's how far they are, this whole region. And this is a 8 units. And there was AD, which is 25 uh, map units. So we need to make A and D, which is 25. So D might be somewhere here if we go in terms. Let's see others as well. So B and C will obviously be 20 because this is 8. And so this order is fine. So it will be obviously A, B, and C because A and C is total 28 and A, B is 8 and B and C is 20. So this is 20. Yeah, so this order is perfectly fine. We need to decide whether D will come here or D will go somewhere else, here or here. So it is 25 units. So A and D is 25. So if we talk here, it will be 25. Then let's look at the distance of B and D. So B and D is coming up as 33 units. So it will be obviously this side of the that specific gene, which should be this side of the chromosome. So this is how the pattern should be D, A, B, C because this is 25, then 25 plus 8 becomes 33. So this whole thing B to D is 33. So that's how we can determine the sequence of genes. So basically we try to determine map units here first and we can, so this is how you can solve this certain question. So it should be D, A, B, C. So the answer is D here. So that's how you solve this certain kind of questions. So let's check the other one. Uh, so this is basically talking about all of this itself, like how this chromosomes are changing and how the alterations happens. So and so basically whatever changing is happening. So initially, if there are two genes involved, how far they are, that can be determined using all those techniques as well. So this is just a question for understanding purpose and how we can actually determine. So if someone knows that A and C are these much units apart, and if B and C are this much units apart, then people try and analyze. That's how the pattern of genes might be there in a chromosome. So that's how it helps. And the farther apart, mm, but two genes are dash. So we need to understand whether it is higher or lower as per the options. The probability that a crossover will occur between them. Hmm. So it is apart two genes, and the probability that a crossover occurs between them, and therefore the dash the recombination frequency. So if the distance between two genes, if the distance between two genes is far away, for example, there is one gene which is A and there is one gene which is B, and whichever is the autosome, sorry, homologous chromosome of it, will also have uh, uh, A and B, right? So even the other chromosome will also have A gene and B gene. So if they are far enough, so generally if the crossing over will happen, the crossing over will happen in this specific location itself. So only this will be getting So the other part of the gene will not be getting switched. So only this part will be getting switched. If the recombination is happening at this specific location, then this will be getting switched. So here only this part of the this sequence of the chromosome is getting recombination is taking place. So if they are apart, so if we look at this, so if further apart two genes are the if they are higher, like then the probability that a crossover will occur between them, and therefore the recombination frequency will also be higher. So that's how you can solve this. And in terms of other questions, so chromosomal abnormalities occurred due to missing extra or irregular portion of chromosome DNA. Which of the following would not disrupt genetic balance? So, so we are talking about chromosomal abnormalities due to missing or extra or irregular portion. If you see these are trisomies, so there is extra something which is there in terms of these two options. And in terms of aneuploidy as well, that is either irregular or missing. It can be anything in terms of aneuploidies. But in terms of polyploidies, it is nothing like that. It is uh, fine enough. So which of the following would not disrupt genetic balance? So only polyploidy is the one which doesn't disrupt the genetic balance, whereas all these three can disrupt. So let's there were a few questions as well to in order to solve them. So in some dogs, the genes of this specific gene, which is for multicolor, is co-dominant. It's not dominant. So it indicates that both will be expressed. That is co-dominance. And a female that receive a B and a W gene have a blank and white spots on a brown coat. So these are we are talking about dogs here. So there is a brown coat on and there is black and white spot. And males can have a black or a white spot, either black or a white, whereas these has black and white, whereas this has black or white spots, but never in terms of multicolors. So so in terms of this, so when a female genotype would look like something like this, which is BN. So this is the female wherein it is having B as well as W gene. That's the reason they are having black as well as white. So B is black, W is white on a brown, like it has a brown coat, and on that there are spots. So a cross of a female black dog. So there is a female black dog with a male white dog. So what percentage of pups will also be calico? So this is calico and this is multicolor, which is codominant, wherein black and white both will be seen. So they are talking about female black dog, means it has doesn't have B as well as W, black as well as white, so it has only white. So that's the reason we are taking a female black dog and as well as a male white dog, indicating one gene will be W and other is Y chromosome. So we are only talking about X-related genes. So when you cross them, when you make a Punnett square board, you can easily get the answer of how many pups will be calcium, indicating both the genes will be expressed. So there is one with PW, there is one with PW. So there are two, so out of four, there are two chances, indicating there is a 50% chance. Similarly, a red-green color blindness is caused by sex-linked recessive disorder. We already looked about it. It is an X-linked recessive disorder. A colorblind man marries a woman. So there is a colorblind man. So this specific man is colorblind. So we have taken that. So that the star indicates that a sex-linked recessive allele. So it is an X-linked recessive allele. And so man is a colorblind and a woman with a normal vision. So woman is a normal vision uh, whose father was colorblind. So his father was colorblind, indicating that female is a carrier. So what is the probability that first son will be colorblind? So we need to look at the sons itself. So if you look at this, so we don't have to deal with females here. We need to deal with son itself because they are talking about their first son. What is the probability of first son will be colorblind? As per the question, we can clearly see that the man is colorblind and female will be carrier because uh, it's so the question was but his that female's father was colorblind. The woman with normal vision, but father was colorblind, indicating that she should be a carrier. So because her father was colorblind, her female is a carrier here. So if we cross them after making a Punnett square board, you can clearly see that there is a chance of two male childs wherein half of the child can be a colorblind and half will not be having specific disease. So that's the reason there is 1/4 of for each daughter, but the question is for only son, so it will be half for the first son. So this is your major answer, but you can also talk about females as well. So there is a 1/4 chance for each daughter. Similarly, there was one more question related to hemophilia, wherein so if we read the question, so she marries a man who is normal, whereas a daughter of a normal phenotype. So daughter phenotypically is normal. So daughter marries a man with a normal phenotype. So daughter is marrying someone with a normal phenotype. What is the probability that a daughter of this mating with a hemophilic and that a son will be hemophilic? So if it is crossing with a hemophilic one, what is the probability that a son will also be hemophilic? And if the couple has four sons, what is the probability that all four will be born with hemophilia? So now we initially have to determine that what is the percentage of sun that it will be hemophilic, and we can just multiply that into 4 and repeatedly, then we can understand the probability of that as well. So man was colorblind and daughter was carrier of it. So when the daughter marries with a normal phenotype, so this carrier marriage with a normal phenotype, you can get this cross and you can understand that females will be carrier and male can be either without the disease or with the disease. So this is the only son and this is the sun. So if you can see half of the chances is for, so the sun will be hemophilic, there it is a half chance. And but if there are four suns, then you can just multiply that half 4 times and you can get the answer that is 1 by 16. So these are the answers for it. So a daughter of this mating will be hemophilic. So daughter cannot be hemophilic. You can see one can be carrier and one is not affected at all. So that is 0. And whereas in terms of others, question the son will be hemophilic. There is a half chance that a son will be hemophilic. And in terms of four sons, we can just multiply with all of them. The third answer will get, which is 1 by 16. Okay, so assume that genes A and B are linked and there are 50 map units apart. So an animal which is heterozygous at both loci is crossed with one that is homozygous recessive at both loci. So what percentage of offspring show phenotypes resulting from crossovers? So the answer is 50%. You can, if you just assume that genes A and B are linked and so they are 50 map units apart, so they are far enough. And an animal heterozygous at both loci. So it is an heterozygous at both loci. So if you look at the solution, so these are the two genes A and B, and so we need to determine the offspring percentage which is showing phenotype after the crossover. So if you just where am I? Where am I? Huh, okay. So you can get the answer as 50%. You can clearly see that these are two parental type and these are two resulted from a crossover. So two genes of a flower, one controlling blue. So there is one uh, of a flower itself, there is one gene which is controlling blue and there is one which is white and other controlling round as well as oval. So it is similar to that of yellow round and green wrinkled. So here blue colored with round shape and white petals versus oval stamens. So it is about stamens and flower color. like petals and are linked at a 10 map units apart. So they are 10 map units apart. These two are. The resulting F1 progeny are crossed with homozygous white oval plants. So there is white petals, which is recessive, as well as oval stamens. So recessive plants were crossed, and 1,000 F2 progenies were obtained. Then how many F2 plants of each of the four phenotypes do we expect? So if you look at the solutions of it, so there is homozygous blue oval plant. So if you see, they are homozygous. So oval is small r and blue is capital blue. So blue oval, so capital blue both. And so as they are recessive, we need to accordingly have those genes. Then we were told that it is homozygous and homozygous with a homozygous. So all are homozygous. It will not be a carriers. So both are dominant. So this is blue oval, this is white round. When you cross them, they will be heterozygous, but they will be having similar phenotype. So when they are crossed with each other, this specific F1 generation, then we can get the F2 generation, wherein now we know that recombination frequency is 10%. Then that's the reason will be whatever is the individual, 1,000 individuals, so half of the individuals are blue oval, half are white round. Then we will accordingly multiply with the 10% of it so that we can understand the recombination frequency of it. So once we do that, we can understand there is a 50% chance there will be white oval uh, kind of plant. So these are the recombinants. So there is a 50 number of as there is a chance of recombination of 10% because they are apart by 10 map units, so indicating there is a 10% chance of getting crossing over or basically recombination. Then if these are 50-50, then accordingly uh, uh, these will be, as if you can see, they are divided in a 50% chances, out of which 10% there might be recombination chance. So accordingly, removing that 10% blue round and white oval, and whatever are the parental characteristics, blue oval and white round will be maximum, blue oval and white round, and the recombinants will be having a different characteristics itself, which will be blue round, like blue round and white oval, which will be opposite to the parental observation. So that's how we can determine the answer. So in order to summarize the second lecture, we are we can see that the linked genes tend to inherit together because they are located near to each other. If there is, so if you can see here, so majority of the individuals were of the same character, but as they were, there is a very minimum chance of getting crossed over because they are just 10 map units apart. There is only 10% chance that recombination can take place. If that is happening, so in this 1,000 individuals, there is only 10% chance, which is about 100, and there are two characters basically. That's the reason we are dividing first into dividing by 2, and then we are considered only 500, and the recombinant chance accordingly are divided. So recombinant offsprings exhibit new combination of traits inherited by two parents, so it will be having new combination of traits. So that's what we are looking at here. And because of the independent assortment thing of chromosome, unlike genes, exhibit 50% frequency of recombination. So there is a 50% chance. So that's the reason we are considering, not considering a 1,000, we are considering as 500. So there is a 50% chance of frequency of recombination in gametes. And similarly, for further summarization of it, for linked genes crossing over between non-sister chromatids, in the non-sister chromatids in the sense the one which are not homologous, during meiosis, one accounts for observed recombinants always less than 5% of the total. So all these numbers are coming into picture after several research and people tried to understand how the inheritance pattern happens. And the order of genes of a chromosome and a relative distance between them can be deduced from recombination frequency observed in genetic process. So this the order of genes, as we already talked about that. So this specific order is actually understood after the number of offsprings, after number of crosses happening, and what kind of offsprings are getting to visible. So here, if we are getting out of 1,000, you might be getting 50-50 of the recombinant characters, indicating that there is only 10% chance of it, so which indicated that these two genes are 10 map units apart. So that's how people try to uh, understand like how far two genes might be, like if you are trying to study those two specific genes. So in order to conclude the lecture of the second lecture, the recombination of unlike genes and independent assortment of chromosome was observed, and the concept of crossing over and the recombination of linked genes. Linkage between two genes affects inheritance of characters based on Morgan's experiment, which we talked about, and few chromosomal abnormalities, which we also talked, like what kind of diseases happens and how alteration of chromosome number and structure cause some genetic disorder. We talked about Down syndrome, we talked about Turner syndrome, we talked about Edwards syndrome as well as Patau syndrome. So you need to remember like what chromosome is getting affected. So can a genetic trait be transferred between different bacterial strains? So you all might be knowing that there is gram positive, there is gram negative, there are different species of bacteria, etc. So can that be transferred? So that is Streptococcus pneumonia. One is a smooth strain, like presence of a capsule, which causes pneumonia basically, and there is one rough strain which doesn't have a capsule, which doesn't cause pneumonia. So, so there were two strains available. So if it has the capsule, it is causing pneumonia. If it doesn't have, then it is causing then it does not cause pneumonia. So if so, people try to study that can that genetic trait, like if the capsule is given, can it also transfer, like it can be transferred to other bacterial strains, and can it also show disease causing stuff? So this specific experiment was done by Griffith, and they actually tried to study the transforming principle. What is that which is going from one individual parent to the next generation, and what is exactly uh, transferring, whether it is DNA which is transferring, whether it is because of the proteins or whether it is because of the RNA. People initially didn't know what is that transforming factor. Like, is it happening because only of the DNA transfer, or like what is the main? Initially, the genetic material was not known. So even Mendel didn't call it as a gene. Mendel called it as a factor. So what is, so people did many more experiments to understand and like basically to prove it. Like some people during that uh, era of research, people believed that it might be protein because everywhere it is the strength or the major building block of our body itself is protein. So proteins might be the one who actually are transferring the message from one generation to another. So many people believed that it might be protein, and there are many who believed that it might be DNA. So there was a debate until and unless people proved that it was DNA itself. So this was one of the experiments where people Griffith tried to understand what is the transforming principle. So in this experiment, what he tried to check was, so as we already studied, in just a minute, okay? I hope you all are there. I'll just, yes, I hope Unity, you're getting it, as well as Pratiksha. Thanks. Okay. So, if you look, so what was the transforming factor? So people tried to understand that. So in this specific experiment, what Griffith done was, so he took a bacteria which was having a capsule, so which he called it as a smooth cell, whereas the one which didn't had a capsule, he called it as a rough cell. So it is a living S cell and a living R cell, whereas when the thing was known that this smooth cell are able to cause pneumonia, whereas these rough cells are not able to cause pneumonia. When it was injected onto mouse, this specific bacteria, it found that it caused pneumonia and mouse died. Whereas when the rough cells were injected, this couldn't cause any disease and mouse was healthy and surviving. Similarly, what was done is there was a heat killed so basically this specific bacteria were taken and they were heated and just so because of the heating, so initially they didn't know whether heating will actually kill the ability for it to cause the disease. So heat kill was taken and it was injected into the mouse, and Griffith observed that it was not showing any kind of disease and mouse was healthy, indicating that by heating, the ability to cause disease or the capsule might be getting degraded and it is losing the ability for cause the disease. That was the conclusion after this experiment. Then we moved on to mix these two specific bacteria, one the heat killed one, the heat killed S cells, and leaving R cells. So this is heat killed. So this is inactivated any which way, then it cannot replicate. It is not showing anything, but it can. But when they mix these two, then it was fascinating to see that this was heat killed already and this was a rough cell and which is not able to cause any disease when it is injected separately, but when it is injected by after mixing it actually causes the disease. So what Griffith tried to conclude out of it is that there is some factor which might be getting transformed by the S killed into this living cell and it is gaining the ability to cause the disease. So what was that factor? He further did experiments to understand that. So here he actually proved that there might be some factor which actually which is actually allowing the R cells to gain that ability and cause the disease because that was living and this was inactivated in which was. So is it protein or DNA? So people were confused, and the genetic material of FAST-T2, whether it is protein or a DNA, and he tried to understand that. So these were the two scientists, Alfred, like Hershey and Chase. It's known as Hershey and Chase experiment. And so what they have done is, so in terms of protein, as you all know, it has sulfur content, whereas in terms of DNA, it has phosphate. That's what like gives the charge to DNA. So DNA has phosphate and protein as sulfur. So what they have done, they use radioactive sulfur and radioactive phosphorus to trace the fate of protein and to trace the fate of DNA. If it is getting transformed, if it sulfur is visible in the next chain, indicating that protein is getting transferred. Whereas if it is phosphate will be seen, then it is indicating that DNA will be getting transformed. So just to and with the radioactivity observed in the next chain, we can understand that whether it is protein which is going or DNA which is going. So such kind of experiments were done in order to understand whether it is DNA or a protein. So uh, so that was this radioactive labeling was done with the bacterial cells and they were pelleted down and the transformation of with the phage was done. So when that was done, so in terms of how exactly that was discovered, so the bacteria is growing and that specific bacteria was given radioactive sulfur treatment for protein. Huh. So all the proteins which are produced by this specific bacteria had a radioactive sulfur. Now a radioactive protein bacteriophage was infected to the bacterial cell. So these are bacteriophage and this is a bacterial cell and this radioactive bacteriophage are infected. And when these were infected, along with there was a control with empty protein shell, and this is the phage DNA which went into them. But you can see there was a sulfur, and when they tried to understand the radioactivity of it, so radioactive phage protein in the liquid. So in the liquid, it showed the sulfur, indicating that all the proteins were outside, only DNA was inside. So pellet of the bacterial cell that contains the bacteria and the phage had all the protein and only that was radio labeled in the liquid, whereas the pellet didn't show any radioactivity, indicating that nothing is protein are not getting transformed into the cell. So it was DNA which was getting transformed. Similarly, phosphate was done, and they tried to understand that at that time this was not getting radio labeled, whereas these were getting radio labeled, indicating that it has been transformed into the bacterial cell, and that's how people understood it is DNA which is the transforming factor and not protein. So this we already studied in our initial first or second week lectures, how DNA came into picture and how these were the scientists who actually predicted the structure of DNA, that there are two bonds between T and A and there are three bonds between G and C. How is that structure? We dealt with all of that in the first week of lectures. And there were experiments done how exactly DNA replication takes place. So DNA with the isotope of nitrogen was done. So wherein if the replication, so one of the DNA was labeled with 15 N, which is an heavy isotope, and other bacterial strain were uh, given a lighter isotope which is 14N. So 40 lighter isotope which will be less than, but indicating that it will be after centrifugation it will be above, and the one which had a heavy isotope after centrifugation it will be comparatively below. So DNA sample centrifuge after the first application, so this is how, so they were mixed together, a bacterial culture in a medium with 15N isotope and bacterial transferred to the medium with. So this was the initial culture and this was transferred to the medium with 14N lighter acid. So basically for DNA replication, it will try and utilize nitrogen, right? And there is a nitrogenous base in in DNA, right? So that's what the structure we were talking about. So that nitrogenous base will have nitrogen in them, and all of those will be labeled with either of the isotope, and the new nitrogen which is available is of uh, lesser isotope, whereas the older DNA, so what generally, as now we know how exactly replication happens, it gets split and the new ones, the new nitrogen source which will be available for it to get replicated, then one will be added here in the other strain and one will be added in the other strain, so indicating that one will be having, the older one will be having 15N nitrogen isotope, whereas the new ones which are getting added, because in this specific media there is in this that medium there is lighter isotope source, so accordingly that will be added in the next strain, right? So people try to understand how exactly that replication happens, and people predicted either it can be dispersive, semi-conservative, or conservative, but it was later understood that it is in a conservative manner itself. So this is light blue is of 14 and dark blue is of 15 and that's what. So there are different ways people thought about it, and people came to know that most of the replication happens in a semi-conservative manner. So this 15 and get split up, and whatever the 14N source was available, that using that source the other strand of the DNA is generated, and in the next replication these two will be separated, and accordingly the next new 14 N using 14N as the nitrogen source, uh, the DNA will be replicated. That's how DNA replication happens, which is semi-conservative manner. So this was predicted basically depending upon after centrifugation, and people tried to see that it is coming in between and accordingly indicating that 14N and 15N are getting together, because it is neither coming at 14N position, neither at 15N position, but both were together in the double stranded DNA, indicating that both might be there, and that's how replication might be taking place. So that's how people understood it is in a semi-conservative form and not in a conservative form, neither in a dispersive model. So people concluded that DNA replication happens using semi-conservative method, and it was a simple experiment using two different isotopes, 14N and 15N. So Mendel's law of inheritance were in the first few lectures. Morgan tried to provide the chromosomal basis of the Mendel's law itself, and classical genetic experiment like if, DNA is the one which is the factor which is transferred across the generation was done in these lectures. So now we kind of know a broader picture of how inheritance happens. So initially we started with in the earlier lectures as well with the Mendel and how happening because of mm, Morgan tried to prove that it is happening with the help of uh, it is in there is an involvement of chromosomes and so people try to prove that, but still it was not proven that it's only DNA and not protein which is getting transferred, and people came with certain experiment like for which we talked about, like it's the DNA which is the transforming factor and not protein. Then in terms of in the next lecture, sir, I talked about bit of bacteria, like how they look and how what do they eat and how do they move, etc. and how do they reproduce. But how can we distinguish between a gram-positive and a gram-negative bacteria? So that happens with the help of its cell wall that we'll be talking about. But before that, to give a broad overview of what prokaryote is. So prokaryotes. One of the major example of prokaryote is bacteria. So they have considerable genetic variation and they are have rapid reproduction. They have mutation possibilities. They can do genetic recombinations as well. And it allows researchers, because of all of these properties, it actually allows researchers to investigate molecular genetics in a simplest true organism, whereas in terms of we are very much complex as compared to that of bacteria. And in terms of their chromosome, it is a circular DNA molecule with few associated proteins. And they also have extra chromosomal DNA, which is called as plasmids, and which replicate independently and they don't depend upon the main genome. And so these are the few characters of bacteria, and along with that, bacteria reproduce rapidly. Therefore, new mutations can be uh, uh, because of the rapid division, the mutation chances increases, as well as further genetic diversity can be arised by recombination. So in the meiosis part of bacteria, when it is dividing, because of that, there can be recombination chances, and that's how mutation rate much more increases, and the speed of division also leads to mutation. So in terms of their cell surface, like gram-positive and gram-negative, we already studied about its cell walls. But how do you distinguish between them? So that happens with the help of staining. So gram-positive stains take purple color and gram-negative takes pink color. And that is because of the phospholipid bilayer present in this specific bacteria. So how do we do this specific experiment is we take um, two different dyes, that is saffronin and crystal violet. So initially we stain the uh, mixture of bacterial samples using crystal violet first. So if you have a bacteria sample and you don't know whether it is gram positive or gram negative, this is what procedure is done to understand what kind of bacterial sample you are having. So initially, crystal violet is added. So now this is taken up by both gram positive as well as gram negative bacteria. Uh, and then iodine is added. So wherein with the help of iodine, which is helps the crystal violet stain to get attached with the gram positive bacteria only, whereas it is not able to attach the gram negative bacteria. That is because of so now with the help of iodine addition, the interaction between crystal violet and iodine and with the cell membrane of a gram positive bacteria helps them to be fixed basically. And when we do alcohol washing or decolorization, at that time gram negative bacteria crystal violet gets washed away. So here you can see the gram negative as well as gram positive both were stained. And when you add iodine, the stain gets fixed in the gram positive bacteria, whereas in the gram negative bacteria, it is not fixed because the cell membrane doesn't support that fixing. And when it is washed away with the alcohol, ethanol, at that time, gram negative bacteria become colorless. And a counter stain is added, which is safranin, which then again observed by gram negative bacteria, and that's how we can differentiate between a gram positive bacteria and a gram negative bacteria. So this is a simple experiment which was discovered by Gram itself, and that's Gram who was a scientist, and that's the reason this experiment is labeled as Gram staining. So because of the specific cell structure, cell wall structure, a gram-positive bacteria can take in crystal violet, whereas gram-negative bacteria is not. It can take in, so it can also get stained, but it is not fixed. But it can easily be washed, whereas with from the gram-positive, it cannot be washed. And how do we exactly kill these bacteria? That is with the help of antibiotics. And so there are few antibiotics which deal with killing that specific bacteria by targeting their cell wall, and there are few antibiotics which deal with the DNA replication part. There are few antibiotics which deal with the transcription part. There are few antibiotics which deal with the translocation part or the protein synthesis part. So accordingly, like each antibiotic has their own specific target where it targets so that it kills the specific bacteria. In terms of penicillin or vancomycin, this specifically inhibits the cell wall synthesis. So basically, they are targeting the cell wall of the bacteria. So once it targets that specific region, so the cell wall synthesis doesn't happen, and when it is not happening, the cell is unable to survive and it gets killed. So basically, there are two terms for it. It is either called as biostatic or not biostatic, bacteriostatic or bactericidal. Bacteriostatic indicating that there are certain antibiotics which will just suppress the growth of bacteria, whereas bactericidal indicate that it will kill the cell. So these are examples of killing the cell, which are bactericidal, whereas stopping DNA replication or stopping protein synthesis, all of these are suppressing the growth. They are not killed directly. The growth is suppressed because there is no DNA replication happening or there is no protein synthesis happening. So these specific antibiotics are called as bacteriostatic, and these are called as bactericidal antibiotics. So they bacteria have both beneficial as well as harmful impacts. There are few bacteria which are pathogenic, there are few which are non-pathogenic, and so they play a major role in recycling chemical elements between living and non-living. Some act as decomposers, which break down dead organisms, and there are some which are helping the plant to have more availability of nitrogen, phosphorus, and potassium. And there are some which are having symbiotic relation with larger organisms. So it can be either beneficial as well as the one which are pathogenic or harmful to us, which cause different diseases like Salmonella typhi, which causes typhoid. There are Asorius, which causes a number of different diseases. In Asorius itself, like Staphylococcus aureus, there are some bacteria which are non-pathogenic and some which are pathogenic. Which one of the disease which is coming into my mind is UTI, urinary tract infection happens because of that specific bacteria, and there are others also which are involved. And so these are experiments using prokaryotes which have led to important advances in DNA technology. Uh, so which we talked about. And another example is Agrobacterium tumefaciens, which is used for to produce transgenic plants as well. So basically, people use that specific bacteria to transfer not the plasmid but the whole bacteria itself to transfer certain genes from one organism to another. So here, plant is used to transform them. So it can be used for making fertilizers, it can be used for making ethanol, etc. So that was about bacteria. And in terms of viruses, so how small they are, that's what it was depicted in terms of this specific image. And so very small infectious particles with genetic material and a protein code, which are nothing but viruses. And they are obligate intracellular parasites, indicating that they require an host cell to reproduce. Outside the host cell, they cannot survive. In fact, almost all members of all three cellular forms of life, bacteria, archaea, as well as eukarya. So they basically these are acting as the host for viruses. They can only replicate inside the organism, not outside. And in terms of the revolution or extremely large DNA viruses, there is Vaccinia virus, which was discovered in 1796, which had 190 kb genome. Then there was Pandora virus, there is July as well, there is Mimi virus, which is discovered in 2003, which had a 1.2 MB genome, which is very huge, whereas this had 2.5 MB genome. So, viruses contains a nucleic acid surrounded by a protein coat, so which actually causes different diseases. So this is tobacco mosaic virus. TMV is tobacco mosaic virus, which actually stunts the growth of tobacco plants and give their leaves as a mosaic shape. So that's the because of the kind of disease this virus causes, and that's the reason it has a name called as tobacco mosaic virus because it gives a mosaic structure to the leaf. And there is something called as bacteriophage as well, which are nothing but the viruses which can infect and set in motion for to take over the bacteria. So it's basically a virus which can infect the bacteria, which are called as bacteriophages, such as E. coli, one of the bacteria. So those are also viruses, but they can infect bacteria, which are called as bacteriophages. And in terms of the structure, they are made up of DNA or RNA. Most of them are RNA, but there are few viruses which has DNA as a genetic material. They have a capsid, which is generally majorly made up of proteins, which that's the reason it's called as protein shell. And there are some viruses which have envelope as well out of that protein coating, and it is a membranous covering derived from the membrane of the host cell itself. So when it is infecting the host cell, when it is coming out via exocytosis or by cell lysis, the outer coating is of the cell membrane itself and of the host cell. So this is one of the example of bacteriophage, which is T4. It is called as T4 bacteriophage. It has a complex capsid containing an icosahedral head. So this is called as head of the bacteriophage, and this is the tail apparatus of the bacteriophage, and it can sit on a bacteria. And so these are bacteriophages, and this is the bacteria. So bacteriophage is the one which virus which can infect the bacteria. And Sir has talked about a bit of RNA viruses. So retrovirus is an example. So one of the example is HIV itself. So it has RNA, which is a positive strand RNA, and they can be integrated into the host genome as a provirus. So that's how HIV is infected into the uh, host organism. So if you talk about HIV, they majorly target T lymphocytes and uh, basically target the CD4 receptors of the T cells, and that's how it by binding to that receptors it goes into the cell and it releases the its genetic material. It also has reverse transcriptase in its genome, and that's how it gets converted to cDNA and gets multiplied. It again makes the whole structure. There is an assembly of the virus and then it releases. That's how it infects in terms of HIV. And there is one more virus which is polio virus, which actually invades the nervous system, causing paralysis in over 200 children. And later people came up with vaccines for it, and now children are not affected with it. So the polio vaccine, which are generally given to all the children. And SARS is an example of COVID as well, causing agent of coronavirus. What is SARS? Basically, it is severe acute respiratory syndrome. It happens like one of the virus which can cause SARS is coronavirus, and this corona word comes because it appears like a crown-like appearance, like it has spikes which are made up of glycoproteins That's the reason the name is coronavirus. And some other viruses which deal with like infect plants. So this is one of the example for it. And Sir has also talked about swine flu, which is a respiratory disease. So what is swine flu here is, so it's SISOIV, which they are called as. So its origin is H1N1 influenza virus, but with the swine, so swine indicating pig basically. Uh, so it is a respiratory disease of pig caused by type A influenza viruses. Uh, And these are certain symptoms of it, and mainly from a person to person, and it is contagious just by sneezing, coughing, it can get transmitted. In terms of influenza virus, they are having hemagglutinin and neuraminidase. So these are the two enzymes which actually help the virus to uh, go inside a host cell. So it actually binds with sialic acid present in the cell membrane of the host cell, and it allows the this binding of hemagglutinin with sialic acid actually helps the cell to go inside the host organism, and once it goes it can replicate inside the host organism, and in order to come out it breaks the bond between sialic acid and to come out of the cell with the help of neuraminidase. So that's how H1N1, basically the name is H is hemagglutinin and N is neuraminidase, and these are the major enzymes involved in taking in the cell, replicating stuffs, and making few more copies and coming out of the cell and infecting few more newer cells. So there is a risk of generating novel viruses through reassortment, and resistance against inhibitors is also possible, making bacteria much more mutated and much more infective. And scaling up for the mass production of vaccines is challenging because it is not easy to produce vaccines, and scaling up will be much more difficult. And these are currently certain challenges, and people are trying to come up with certain solutions for all of these challenges. And there is a rapid reproduction mutation rate in terms of prokaryotes. That's the reason they are becoming much more intolerant to antibiotics. And Because of that, people uh, are into medication, and even if you and that is also happening because of overuse or misuse or overdose of antibiotics there because of certain negligence of individuals itself. Bacteria are gaining mutations and they are able to survive antibiotics. And even if a specific individual doesn't complete a course of antibiotics, and there are some few bacteria just adapted to that and got mutated, they try and replicate. And if they are getting transferred somewhere, that's how the mutation rate is increasing in terms of bacteria. And these were the four lectures which Sir talked about, and there was a talk by one of the professor in the fifth lecture. Uh, so you can look at that talk as well. It was a great talk, and that's it in terms of revision. And we can look after certain previous year questions. We can take a break of maybe like 2-3 minutes, and we can start with the questions. Okay, we can start with the questions of previous semester. Okay, so now if you look at this question, so consider a family wherein we are talking about most possible inheritance pattern of this disease. So, so one there is a male who is phenotypically normal and female which is also phenotypically normal. But if you look at this specific outcome after they were crossed, you can see there is a chance of majorly there is a chance of being phenotypically normal, but there is a disease state as well. So disease state indicating that these are two individuals are carriers probably because uh, uh, there is a disease state as well. If they were dominant for this specific whatever disease they are talking about, so there is they have not talked about what kind of disease it is, but we need to talk about most possible inheritance pattern, whether it is autosomal dominant, autosomal indicating that there is nothing to do with male or female here, and if it is if you get a observation regarding like daughters are affected or only sons are affected, then we can think about this specific Y-linked dominant or X-linked dominant. So if male would have been affected, then only males would have been affected. Then it indicated that Y-linked dominant. And if that was the case, then this specific male parent should be affected and not phenotypically normal. So this should be eliminated. Similarly, X-linked dominant. If that's the case, then this parent should also be affected or a carrier at least. So it like looking at the outcomes here, it should be majorly either autosomal dominant or autosomal recessive. But you can see majority of if it was dominant, like there is a disease state as well, so indicating that they are carriers for sure. And if it was dominant, the carriers would have been showing the effect, indicating that they would be also disease. So this indicates that the answer should be probably autosomal recessive. Let's look at the answer. Yes, it is autosomal recessive. So that's how you can like think of or eliminate options and you can come up with an answer. So now here, this is so they have already informed us that the question says that it is about color blindness, and we already know that it is a X-linked recessive disorder. So females can be carrier, whereas generally males will be affected because X-linked recessive disorder it is. And what will be the so it is a recessive disorder. You can need to consider this as well. And what will be the chance of having a colorblind male child to this couple? So they have given two generations already. So there was a colorblind grandfather. So one X is coming from here for a female, and there is a colorblind. So there is a son here. So they have a son here, and this specific man actually got married to a colorblind carrier mother. So uh, she is carrier for sure, and whether this specific man is phenotypically normal. So there was colorblind father indicating that X. So for from a father, generally a Y chromosome will go to a generally not always a Y chromosome will go to man like male person, whereas the X will be coming from uh, the female. So even this grandmother was carrier though, but if this individual was phenotypically normal, indicating that he is not carrying the gene, the Y is coming from the father, whereas where he got married with a carrier one, so indicating that a normal female, normal male you can just X, you can take this as X and Y, and this you can take as X with a carrier sign of color blindness and X with no sign. So when you cross them, so you need to cross this specific character, then you can easily get the answer, which would be 50%. And we are talking about male child itself, so whatever Y will come. So one of the X will come by this uh, female. So one can be carrier and one will not be a carrier, right? So that's how XS will be splitted up in terms of this. So as one X will combine with this Y and one X will other X will combine with the other Y. So one X is with the carrier, so half of the chance will be for colorblind and half of the chance will be for normal. So the answer should be 50%. Okay, so so that is a cross between this. So here genes are involved with body color and eye color. So the golden bodied red-eyed one are normal ones, whereas the gray-bodied, gray-colored eyed are double mutants. So we're talking about body color as well as eye color. So they are double mutants, not having the body color as well as the eye color, whereas these are dominant. So gray and red-eyed, whereas these are golden-bodied and red-eyed, which is completely dominant. And when these were crossed, it obviously generate a mutant, like carrier of it. So phenotypically it looks the same, but it is a carrier for both gray-bodied as well as. So this genotype we know. So we need to determine the genotype of this if this is the outcome. So now if you look at so let's look at one specific character at a time. So there is G and G plus. So if you can see here, there is G and G plus. So indicating that this specific, so one G will come from here and one G will come from here, right? So if it has G and G plus, indicating that, so if one of the G is coming from here, there might be G plus here as well. And in this specific uh, organism. So if G plus is coming from here, then this should be having G and not the dominant character. But if you see here, there were two chances that this specific individual, specific offsprings are having two Gs, indicating that this should be having two Gs and not having any G plus as such. So this G is coming from here and one G is coming from here and one G. So there are two chances of G plus indicating this is coming from this side, and if there is G like without a plus sign, should be here. Uh, uh, so ideally this should be having gray color itself and not golden color. So this should be an option or this should be an option. So let's see with the eye color. So it is having one R plus and one R. So this is a carrier. So if you see, this was a carrier, whereas this was not a carrier. Similarly, this is not carrier. So if you look at the ratio, there are two chances of not being a carrier. So this should ideally be having two R. So if you just write that specific genotype GG and RR and you cross them, you make a Punnett square board, you will actually get this outcome. And that's how you could solve this question. And your answer is GG and RR, indicating that without a plus sign, indicating that black bodied and black eyed, or gray eyed, I mean to say. Yeah, this is a comparatively easier question. So, which of the following syndrome differs in terms of chromosome aberration from the other ones? So, if you look at all these three chromosomes are depending upon autosomes, that is 21st trisomy, 13 trisomy, or 18 trisomy, whether this is related to sex chromosome. So, it differs. So, this is the only one which is related with the sex chromosome compared to these three. So, that is different. So, the answer should be Turner syndrome. Yeah. So, now we have already solved this kind of questions with the examples which sir taught. So suppose these are certain genes like alpha, beta, gamma, delta, etc. So now, so this alpha and beta are 20 map units apart. So here the sequence is not the same. So arrange the location of genes on the chromosome based on the following recombination frequency. So recombination was performed and according to the number of uh, offsprings observed of that specific characters using those two specific genes, people have come up with the map units of it. So now we know the map units, but so the question is to arrange this specific uh, genes in a order, in a sequence. If alpha and beta are of 20 units, and uh, epsilon and beta, so these two are only 13 units. So this shouldn't be this far. So these two are only 13 units, whereas these two are 20 map units. So ideally, these two should be very far. And epsilon and gamma are about 8 units. So we need to uh, solve this question by, okay, let's try to solve it. So we just take alpha and beta. So alpha and beta, as we can see, it is comparatively 20 units. So I'll just make a bigger. So let's take alpha here. And so it is far apart, beta is far apart. So I'll just take at the end for now. Let's see if that works better. So it is 20 units. So this is 20 units. So now just look at the alpha part itself. So alpha and delta is around 7 map units. So if this is 20, maybe it is coming somewhere here. Maybe let's think about that. So this is around 7 units. So we'll just look at alpha, like how, so alpha, beta, we know that is 20. Now we just looked at alpha and this. There is alpha and epsilon, which is 5 map units. And where is alpha else? Okay, alpha, beta, alpha, epsilon. So now epsilon may be here at 3 units, or it can be either here. So it can be somewhere else. So let's look at beta first then. So beta and this is 13. So this should be perfect enough. So alpha and beta are 20 units, and this epsilon and beta are 20. So sorry, these are 13 units. So this sequence should be appropriate enough because it is matching this character as well as this character, and we have looked about this character as well. So this is fine enough. Now look at, so the distance between this and gamma is 8 units. So now it can be either this side, like just, so alpha and gamma, do we have alpha and gamma map units? No. So let's assume that it is on the right side. So this along with gamma is around 8 units. So maybe let's take gamma here, which is 8 units. And now we have delta and epsilon at 27 map units. So delta epsilon, so let's take at the very corner of it. So this is around 27. So this is around 27 units. Now we have epsilon and alpha of 3 units. So let's assume that it can be either this side or this side. So let's take it as this side, which is around 5 units, which is epsilon. And now the alpha is about 7 units. So we have already looked at that. And now these two are around 15 map units. So, and epsilon. So 15 plus 5 plus 7. Is it 27? Like 15 plus 5 plus, yeah. So like this is how the sequence should be. So that's how you can solve this question. So it is delta first, so delta is here only. So I think this is the answer. That's how we can solve this question. And okay, so in terms of this, so there are two statements. We need to understand which one is incorrect. So statement one says there are four statements. All genes are essentially present on the chromosome. Yes. So genes that are located on the same chromosome will always pass together from one generation to another. So genes that are generally located on the same chromosome will always pass. So is it always or sometimes we need to look after that? So the distance between two genes can never be mapped without cloning experiments. Yeah, ideally it should be like that. And all genes are essentially overlapping present on the chromosome. So pick the incorrect interpretation for the molecular biology expression of chromosomal theory of inheritance. So we just talked about that and it should be easy to answer this at that which one is incorrect. So the statement 2 is incorrect. In an experiment, like two genes were assorted independently, which of the following points explain the best about the chromosomal location of two genes? So here we are talking about independent assortment. So which experiment will talk about this independent assortment in a best manner? So they are located in close proximity on the same chromosome. Which two genes we should select in order to understand that the assortment is independent? So crossing over between the genes does not occur. If we take that, then independent assortment can be easily proven, or the genes are either located on different chromosomes or are far apart on the same chromosome. So either it will be far or it should be close, or the location of genes cannot be predicted based on the given data. So this doesn't come into picture. So which one should be in order to understand independent assortment? So it should be far apart so that we try to understand that either located on a different chromosome or far apart on the same chromosome. So that will help us to understand whether two genes are assorted independently or not. So there was an experiment. So this is about replication. 59 and 49 is allowed to grow and pick the correct statement about two strands of DNA molecule of the first generation bacteria. So as we already know, it is semi-replicative, so it should have two, both of them. So DNA semi-conservative, so it should have two. So DNA strands of daughter cell will have different density and they do not resemble the parent DNA strands. DNA strands of daughter cell will have same density as they resemble parent, or will have different density but resemble parent DNA. They will not resemble though, and will have same density but do not resemble parent DNA. So the answer should be, so there are three which doesn't resemble and there is one which they resemble parent DNA. So what do you think? So just if you refresh the semi-replicative concept, you will automatically get this answer. So DNA strands of daughter cell will have different density, but they will resemble the parent DNA. Yeah, so there was this experiment. I think we talked about this in the earlier lecture. So in terms of pair hairline popularly referred which as widow's peak. So if it is inherited as an autosomal dominant trait, consider the following situation. So widow's peak, this we also did in our last lecture. So male had a widow's peak and female has straight hairline. So uh, this is autosomal disorder, autosomal dominant trait. So let's just consider as WW. So there is no XY, XY or XXXX. So don't get confused between sex-linked inheritance pattern and autosomal inheritance pattern. So if you accordingly take those specific characters, like if you just take WW as a positive widow's peak, and if you take small w as a airline, you can just make a cross and you will directly get the answer for it, which will be 50%. So now we did do gram staining as well. So firstly we gave crystal violet. So the initial either it will be first option or last option. Then we uh, add D-stainer peptidoglycan. So there is nothing like peptidoglycan in the experiment. We add D-stainer alcohol. Then we add counterstain. So before that we also add iodine so that it fixes. So that's not written here, but whatever are the options here. So the fourth option is correct. Yeah, so that's it for the lecture today. So we can meet on the 4th of March for the next lecture. But yeah, if you are having any doubts, maybe we can discuss on that. Yes, there were two others initially in the lecture, then uh, Mohammed came into the lecture. I don't know when you joined in, but we we did revise everything regarding the week 5 lectures. And after revising all 4 lectures we sir talked about, and then we went on to understand, like discuss few previous year questions, which will help you all to solve the assignment questions. Yeah, if you have any questions, ask me, or we can meet in the next lecture on 4th March. That is next Tuesday. Yes, Mohammed, if you are having any doubts, you can discuss, or else we can stop the meeting. Okay, thank you. Thank you for joining in. We can meet in the next lecture. If you are having any doubts, you can still take that in the next session. Thanks.